Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the gymnosperms are flowering plants. The pollen grains and ovules are developed in specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. The range in size from smallest Ulfia to tallest tree Eucalyptus over 100 meters. They provide us food, fodder, fuel, medicines, and several other commercially important products. They're divided into two classes, dicotyledonous and monocotyledonous. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation in leaves, tetramerous and pentamerous flowers, having 4 or 5 members in each floral whorls. The monocotyledons, on other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flowers having 3 members in each floral whorls. Next, the male sex organs in flowers is the stamen. Each stamen consists of slender filament with an anther at the tip. Within the anther, the pollen mother cell divides by meiosis to produce microspores, which matures into pollen grains. Microspore mature into pollen grains. The female sex organs in the flowers is pistil. Pistil consists of a swollen ovary at its base, a long slender style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergo meiosis to produce 4 haploid megaspores. 3 of them degenerated and 1 divide to form embryo sac. Each embryo sac 3 celled egg apertures, 1 egg cell, 2 synergids, and 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. The pollen grain, after dispersal from anthers, are carried by a wind and various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma, and resulting pollen tubes grow through the tissues of the stigma and style, reaching the ovule. The pollen tube enters the embryo sac when 2 male gametes are discharged. One of the male gametes fuses with the egg cell known as syngamy to form zygote. The other male gamete fuses with the diploid secondary nucleus to produce triploid primary endosperm called PEN. PEN. Because of the occurrence of the 2 fusions, syngamy and triple fusion, this event is termed as double fertilization. And in event unique to angiosperms, the zygote develops into an embryo with 1 or 2 cotyledons, and the pen develops into endosperm, which nourishment to the developing embryo. The synergids and antipodals degenerated after fertilization. During the events of the ovules develop into the seeds and ovaries develops into the fruits, and the life cycle of the angiosperm is shown in the figure. Let's see the figure here. The microspores, nothing but pollen grains, which is reaching the megasporophyll ovary through pollinations and development of the pollen grains in the stigma, and all reaching the ovary. The both double fertilization is going to happen, and the formation of zygote to embryo to sporophyte. Here you can see that the sporophyte is a diploid generation 2n, gametophytic is haploid. The fusion and the fertilization restores the number, and it becomes the diploid. Here you can see the female gametophyte with style stigma on the top and the base of the swollen one known as ovary, and it also has megaspore mother cells, megasporangium ovules. Here upper one is filament, which releases a microsporangium pollen grains. Again it is all similar one. Pollination occurs, fertilization, and again formation of zygote to a formation of endosperm and embryo. It is a repeated process, less like a cycle.